Okay, hello everyone, welcome back. This is week 8 of the NPTEL course, Experimental Biochemistry. This course is coordinated by Professor Dasgupta from the Department of Chemistry and Professor Dev from the Department of Bioscience and Biotechnology. And I am Beas Mukherjee, a research scholar from Dr. Soumya's lab in the Department of Bioscience and Biotechnology. And in today's class, we are going to talk about enzymes. Okay, so we are going to talk about enzyme kinetics, the different terms in association with enzymes. We are going to talk about different mechanisms of enzyme inhibition, and we'll take a few examples of different common enzymes and we are going to discuss them. Okay, so let's start with today's class. My voice is broken, I cannot shout, so please bear with me. Okay, if you have any questions, you can raise your hand and you can ask me in the class. Otherwise, you can type in the chat box and I'll answer the questions. Okay, so let's begin with today's class. Okay, so let's start talking about enzymes. Right, so enzymes are biological molecules that catalyze biochemical reactions with very high specificity. The majority of enzymes are, yeah, good evening to you too. Okay, let's carry on with the class. Right, so normally when we talk about enzymes, what comes into our mind is that enzymes are normally proteins, right? But interestingly, not all enzymes are proteins. There is a small class of enzymes which are actually RNA molecules. So we are going to talk about them in our upcoming slide. So the thing that we can see here is that majority of enzymes are made up of proteins. However, ribozymes, they are enzymes made up of RNA. Okay, so enzymes play an important role in all biological processes, including metabolism, digestion, then DNA replication, cell signaling, etc. So they accelerate chemical reactions by lowering the activation energy required for the reaction to occur. So this is a very important characteristic of enzymes. Okay, enzyme activity can be regulated through various mechanisms, including allosteric regulation, covalent modification, and feedback inhibition. So we are going to talk about allosteric regulation in today's class. Okay, understanding enzymes allows scientists to unravel the mechanisms underlying cellular metabolism, disease progression, drug interactions, etc. So insights into enzyme function provide opportunities for development of novel therapeutic strategies, diagnostic tools, biotechnological applications, etc. Right. So one of the very interesting disciplines is enzyme engineering. So in industries, a lot of money, a lot of time, and a lot of efforts are put into engineering enzymes that are designed for a specific function. They have a specific selectivity. They are active only over a specific range of temperature. So this is a very interesting discipline, and if any of you are interested in this in future, so you can actually choose this. For this, you have to know a lot about proteins, enzymes, and be have the skills to work with proteins and enzymes, okay, and design proteins and enzymes. Okay, so we'll carry on. Right, so we will start talking about this non-protein enzyme. Okay, so ribozymes are RNA molecules that catalyze chemical reactions similar to protein enzymes. Some of the examples of ribozymes are ribosomal RNA, that is rRNA, and it contains this active catalytic center, which is the 23S rRNA in prokaryotes and 28S rRNA in the eukaryotes, found in the ribosome, which is a machinery used in protein translation, right? So formation of amino acid chain from this mRNA. So that is actually acting as an enzyme. And what is the enzyme? It's actually peptidine transferase. So it basically transfers this amino acid into the chain from the tRNA onto the growing amino acid chain, right? So that is the function. And the catalytic center is actually present within the ribosome. Now certain RNA sequences called introns, they can remove themselves. Now we know that the DNA is made up of exons and introns, right? So and introns are actually the sequences that are not translated, they are not transcribed into mRNA and they are not translated into proteins. So they cleave themselves out, and these are called self-cleaving introns, and their function is RNA splicing. Okay, now RNase P. Now we know about a lot of different RNases, and we know that RNases, the main function of RNases is to cleave RNA. Right, now RNases are of different types. For example, there is RNase A, RNase H, RNase P. Now this P component, this is actually a ribozyme. So this is a non-protein enzyme, and it is made up of RNA, and this processes the precursor tRNA molecules. So for your understanding, a small table is given and all the functions of these different non-protein enzymes, ribozymes, are mentioned. Okay, so you can go through them. Now we can try to solve one small question. Okay, so which one of the following statements about different RNases is correct? So RNase A is a ribozyme that cleaves RNA DNA hybrid. RNase H is a RNA enzyme that removes introns during self RNA splicing. RNase P contains a catalytic RNA component that cleaves the 5' leader sequence from the precursor tRNA. RNase A and RNase H are both non-protein enzymes involved in RNA processing. So which one do you think is the right answer? Okay, so you can read about this RNA splicing. So RNA splicing is also an important topic. In our next class, we will talk about RNA and DNA. Okay, so you're saying that C is the right answer. Okay, we'll look at it. Yes, that is correct. So C is the right answer. Why? Because both RNase A and RNase H, these are proteins. Okay, but RNase P is not a protein. Now, what is the role of this RNase A and RNase H? Now, it's very simple. I'll tell you a simple strategy for you to remember. Okay, so RNase A cleaves single-stranded RNA. Okay, so it cleaves single-stranded RNA primarily after the residues C and U. Okay, and what about H? Now H, the term H is for hybrid. So this particular ribozyme, this particular protein, it cleaves this RNA DNA hybrid. So when is RNA DNA hybrid forming? You can remember during DNA replication, because the primers are RNA, right? But the complementary strand that is getting formed, that is DNA. So this RNA DNA hybrid is removed by primases, if you remember, right? So this particular RNase H is the protein that performs this cleaving action of RNA DNA hybrid. Now it does not remove introns. Okay. Now RNase P contains a catalytic RNA component, that is ribozyme, and it cleaves the 5' leader sequence of the precursor tRNA to generate mature tRNA, and it is a non-protein catalytic RNA. Okay. Now we will go to the next part. So the next part is again something that a lot of you already know. Okay. So these are the different terms used in enzymology. Right, so one of the terms is coenzyme. So one of this very important question is, what is the difference between a coenzyme, prosthetic group, and a cofactor? So how are these different in today's class? That is going to be clear for you guys. Okay. So coenzyme. Coenzyme is an organic molecule which is often derived from vitamins, and they assist enzymes in catalyzing reactions by carrying chemical groups between the enzyme and the substrate. Now there is a very subtle difference between coenzyme and prosthetic group. So as we go on with our lesson, we will look at that difference. Okay. Now for example, NAD plus is a coenzyme. Then we have cofactors. Now normally metal ions are cofactors, but non-protein organic molecules can also be cofactors. Okay, so they are required for proper functioning of the enzymes. Now we will look at the differences in a little more details right after this. Now, for example, Mg2 plus, zinc 2 plus, these are all cofactors. Now, another important term is apoenzyme and holoenzyme. This you guys already know. So, the protein, the component of the enzyme. So, if you look at this, this is the component of the enzyme without the coenzyme or the cofactor. That is called the apoenzyme. And what is the holoenzyme? Holoenzyme is the fully functional part of the protein, right? So fully functional part of the enzyme, so it contains the uh, coenzyme or cofactor, whatever is required for its activity. All of that is there and all of that is bound to it and it is completely functional. Then we will call it a holoenzyme. So holo means whole or complete. Okay. Now active site. Now active site means that, for example, this is the active site of an enzyme. So it is the 3D space in which the side chains of the amino acids are projecting in such a way that, let's say this is the substrate, it is going to come and the substrate is going to change its shape in such a way that it is going to be complementary to these amino acids, these molecular interactions, and it is going to bind and then the reaction is going to proceed. Okay, so the active site of an enzyme, in the unfolded state, the active site can be made up of amino acids from the center of the protein, from the end of the protein, from the beginning of the protein. But when all of this holds together, all of this comes together into one definite space. Okay, so the active site of an enzyme is a specific region or pocket on the enzyme surface where the substrate binds and undergoes a chemical reaction. It is typically a groove or a cleft formed by the arrangement of amino acid residues in the enzyme's three-dimensional space. The specific orientation, chemical groups, and the charge of the residues in the active site make up the catalytic center for binding to the specific substrate during enzyme catalyzed reaction. Okay. Now this determines the specificity of the enzyme. So that is why the active site of the enzyme is very, very important. Okay. Now there are certain very specific characteristics of an enzyme. They are specificity. So specificity can be grouped into different types. We will look at that. Okay. So a particular enzyme is often very specific towards the chemical group or maybe the 3D orientation, the 3D structure of the substrate. There are different properties. We will look at them. Right, complementary shape. So often the substrate, the shape of the substrate is complementary to that of the active site. So now if you're designing any inhibitor, if that is a competitive inhibitor, you will actually make it complementary to the shape of this enzyme's active site. Now interestingly, the enzyme's active site keep on changing shape. So that is where the problems come in and that is where scope for research comes in. Right. So catalytic residues. Now within the active site there are amino acids that directly participate in catalyzing the chemical reaction. These residues play key roles in the enzyme function. What is induced fit? So I told you that the shape of this substrate changes. So upon substrate binding, the active site may undergo conformational change to optimize the interactions between the enzyme and the substrate. Right. So the shape of the active site actually changes and that is called induced fit mechanism. Now, the characteristic of the active site collectively contribute to the efficiency and specificity of the enzyme reactions by providing a suitable environment for substrate binding and catalysis. So what happens in an enzyme catalyzed reaction? See, initially there was this enzyme, the substrate comes and the enzyme and substrate bind together to form a complex. The rate of this formation of enzyme substrate complex is often denoted by K1 and the reverse reaction is K minus 1. Okay, so the rate of the forward reaction, if that is higher, only then the enzyme substrate complex will be formed because this is a reversible reaction. Next, what happens is enzyme and substrate complex gets formed into enzyme and substrate, enzyme and product complex. So then what happens is product separates out and the enzyme goes for another reaction, right? So this is relatively a less reversible step and that it is catalyzed by, it is represented by this K2 which is the constant, and we will look at all of this in more details, okay, in the upcoming slides. Now we have two questions for you regarding enzyme structure and function, okay, and the different terminology that we learned about right now. So let's try to answer these two questions, okay. So which of the following statements is correct regarding enzyme structure and function? So one is apoenzyme is equal to apoenzyme plus cofactor gives you holoenzyme. Coenzymes are organic molecules that participate in catalysis. Active site residues may be distant in primary structure, but they are close in the 3D structure. Enzyme specificity arises solely from cofactors. So which of these do you think is correct? Now coming to the next question. Which one of the following correctly distinguishes a cofactor and a coenzyme? Cofactors are always organic molecules, whereas coenzymes are always metal ions. Coenzymes are organic molecules that transiently bind to the enzyme, whereas cofactors may be metal ions or organic molecules. B. Okay, good. Let's see. Yes, B and B are correct, right? So now you understand the difference. So specificity mainly arises from the protein structure. So it arises from the orientation, the active site basically. Okay, that's what gives it specificity. So we have new members joining. Okay. Now coming to the next question. It is B because coenzymes are organic molecules that transiently bind to the enzyme. That is correct. And cofactor, it can be a metal ion or it can be an organic molecule, but it will bind very, very tightly to this enzyme. Now, when the coenzyme, when the cofactor is an organic molecule, it is called a prosthetic group. Okay, so we will talk about that in the upcoming slides. Okay. Now, this is how an enzyme reaction happens, right? So, initially the enzyme is there, the substrate comes, and what happens is the enzyme, so enzyme reduces this activation energy. So, what is activation energy? That means for this enzyme substrate complex to form the enzyme product complex, a lot of energy is required, and that energy is minimized by this enzyme. Okay, so what enzyme actually does is enzyme increases the rate of the reaction by lowering the activation energy required for the reaction to happen. Therefore, speeding up the process that are essential for life. Okay, right. Now we will talk about something called the transition state. Okay, so what is this? Now so what happens in transition state is initially, let's say this was the active site and let's say this is the substrate. Now you know about the induced fit mechanism where the enzyme changes its shape, the active site changes its shape so that it can bind to the substrate perfectly, and this state of the enzyme is called the transition state. Okay, and often drugs are designed against this transition state because this is the exact conformation of the enzyme at which the substrate binds. So if I am designing a drug that is a competitive inhibitor, it will prevent my substrate from going and binding. Okay. So now enzyme complementary to the substrate. You can see now this picture has been taken from Leninger. Okay, Leninger's Biochemistry. So when you read Leninger book, you will get to know exactly what is there in this figure. Okay. Right. Now we will go to the properties of enzyme catalysis. So enzyme, like everything else in nature, depends upon certain optimal temperature, pressure and other pH conditions. Right. So what is the thing about temperature? Remember that enzymes, they are not very active at very low temperature. They are optimally active at a specific temperature, and then they are again not active at a very high temperature. Why? Because at a low temperature, the kinetic energy is very less. That is why the enzyme molecules are not vibrating faster enough, not vibrating strongly enough to come across the substrate and to form these bonds with the substrate. At very high temperature, the enzyme is getting denatured. Right? It is after all a protein. How stable will it be? Right? Now, interestingly, different enzymes have different optimal temperature ranges. That is why when you read about PCR or polymerase chain reaction, you know that this particular hot spring bacteria, from it we had taken out its DNA polymerase, and this DNA polymerase was active only at 98 degrees, 95 degrees centigrade temperature. Right? So that is its optimal activity. Right. So now enzymes require an optimal temperature and pH for their action. The temperature or pH at which the compound shows its maximum activity is called optimal pH or optimal temperature, and the temperature at pH more than the optimal one again alters the molecular structure of the enzyme. For pH, you know that at different pH, the amino acids will, the side chains will have different charges. That is why the entire 3D structure of the protein will change. Correct? So, generally an optimal pH for enzyme is between 5 and 7. That is the general pH range that we have in nature. Right? But for certain enzymes, the pH is much different. For example, in the stomach, the pH range is around 2. Right? So it is 2 because the gastric juices, all these gastric enzymes, they are also active. Right? So how are they acting then? So they are used to that kind of pH in that case. Right? Because of the presence of so much of HCl in our stomach, the pH is very, very low. Okay. So heat. The increased enzyme energy level of the molecule disrupts the bonds in the enzyme and between the enzyme and the substrate. That is, hydrogen bonds and ionic bonds are disrupted, hence denaturation happens and the 3D structure is lost. But remember, covalent bonds are not broken. Right? Now, that is why the primary structure is retained. Then for cold, the molecules move very slowly and therefore the collisions are less, as I explained, and that is why reaction is not happening. Now these are the small bell-shaped curve that you see. Go through these graphs. Graphs are very important in this particular chapter. Okay, and enzyme is a place from which you will get questions in every exams. So you have to be very, very well versed with this topic of enzymes, especially numericals. Okay, now temperature, you know that bell-shaped curve is it, right? What about pH? Now for pH, it is even a little sharper, right? So if higher or lower H plus in acid or OH minus in alkaline condition is there, it can interfere with the enzyme structure. This we know. What about enzyme concentrations? Initially, when you increase the enzyme concentration, more reaction will happen. But once the substrate is saturated, that means this is the substrate that I have. Even if you give me a lot of enzyme, what will I do with it? Correct. So it will go into a saturation phase. Similarly for substrate, initially you keep on increasing, but enzyme is limited and so it will go into a saturation phase. Okay. Now we'll move on. And what about specificity? Now specificity is grouped into various types. Okay, there can be bond specificity, there can be group specificity, there can be substrate specificity, stereo specificity, optical specificity, geometric specificity, and cofactor specificity. So one of the examples is alpha amylase only cleaves alpha 1,4 linkage found in starch molecules, and alpha amylase cannot cleave beta 1,4 linkage also between glucose molecules found in cellulose. Okay. Absolute specificity, the enzymes will catalyze only one reaction. That is absolute specificity. Now you have to come up with the examples and read the examples. Okay. Group specificity, for example, hexokinase, if you remember, that is there in glycolysis. So it will catalyze a specific molecules having a specific functional group. Okay. Then lineage specificity. So they will act only on a specific type of chemical bond regardless the rest of the molecular structure. Linkage specificity. And then we have a stereochemical specificity. So they will act on only one specific steric or optical isomer. So enzymes are very specific as they bind to substrates with distinct chemical groups only in certain orientations. Okay. So go through a lot of examples. Okay, now we have two questions here. Try to solve these two questions. Which of the following best represents group specificity of enzymes? An enzyme catalyzes only one substrate. An enzyme catalyzes reactions involving substrates containing a specific functional group. Enzyme catalyzes both L and D isomers of the substrate. Enzyme catalyzes all reaction in a metabolic pathway. The activity of most enzyme increases with temperature up to an optimal value and decreases thereafter. The decrease beyond optimum temperature is primarily due to B and B. Good. Let's see. Yes, perfect. So first one, you know that if it is involving a specific functional group, it is group specificity. And the second one is denaturation of enzyme happens at a very high temperature. Very good. Okay. Now we'll move on. Now there is something called the turnover number. Okay, give me 1 second. So what happens in turnover number is it's also called as molecular activity. So this tells us how many maximum molecules of the substrate can be converted into the product per catalytic site of a given concentration of enzyme per unit time. Okay. So the units of turnover number or kcat are kcat is equal to moles of product per second divided by moles of enzyme or second inverse, right? So the number of molecules of substrate transformed to product by enzymes per unit of time is defined as enzyme activity. So all of these definitions are very important. What is turnover number or molecular activity? What is enzyme activity? And what is specific activity? Okay, so specific activity is the activity of an enzyme per milligram of total protein. Now all the formulae are given for you. The same units are assigned to like two different names. Okay, so the unit of enzyme activity is micromole of substrate transformed divided by minute, and that is its unit. Okay, unit of enzyme activity in new international unit, that is cation, which is moles of substrate per second. Okay, so whether it is in unit or it is in cation, you should be able to understand this is the unit for enzyme activity. Next we have specific activity. So micromoles of substrate per minute per mg enzyme. So that is unit per mg enzyme. Right? Specific activity is moles of substrate per second per kg of enzyme. So that is cation per kg of enzyme, and molecular activity once again is units per micromole enzyme, and this is cation per mole of enzyme. Okay. So next we will look at a particular solution to a problem. Okay, so how to solve numericals on enzyme kinetics. So it's very important that you practice these numericals. One of them is solved here for you. Okay, so 10 mg of a particular enzyme, carbonic anhydrase, it has a molecular weight of 30 kDa. In the presence of excess substrate, exhibits a reaction rate of this micromole per minute. At substrate concentration this, the rate is this. So now they are asking what is Vm, what is Km, and what is the K2. K2 is kcat, right? So enzyme substrate is converted into enzyme product complex. Now how are you going to do it? So remember that in the presence of excess substrate, Vmax is equal to this given. Now that is the rate of reaction. So excess substrate means that if you remember the graph, Vmax was plateau, right? At S, that is substrate concentration this, and the rate of the reaction is this. So that means half of Vmax is equal to Km, excuse me. So Km is equal to the value. Okay, so this we are getting from Michaelis Menten equation. So what is Km? Km is the substrate concentration at which Vmax is exactly half, right? And divide Vmax by micromole of ET to find kcat, and that is how you will get kcat, right? So kcat you saw in the previous slide that what was the formula for kcat? Kcat was Vmax divided by ET. So you just know that. And that is how you solve this problem. Okay, so the problems are simple. All you need to do is just understand how to solve them. So that is why practice is very important. Okay, now we have two more questions. They are simple questions. Try to solve them. Okay, Increase in hydrostatic pressure generally affects enzyme activity because pressure alters primary structure of proteins, pressure affects enzyme substrate complex formation and protein formation, pressure converts enzymes into cofactors, pressure increases Km to infinity. And the second question is two enzymes, acha, B and A. Okay, good. Let's see. Yes, very good. So B and A, that is correct. Right, so pressure affects enzyme substrate formation and protein conformation. That is correct. And the second question, the answer to the second question is you have to find out Kcat by Km. Right. So Km is given, Kcat is also given. Kcat by Km for the first one gives you 10 and the second one gives you 8. So that means you know that more catalytically efficient is the first enzyme. Right, enzyme X. Now, we have another two questions. According to the modern theory of enzyme catalysis, enzyme accelerate reactions primarily by increasing the equilibrium constant of the reaction, stabilizing the ground state of the substrate, stabilizing the transition state of the reaction, increasing the substrate concentration near the enzyme. Which type of inhibitor typically shows the strongest binding to an enzyme? Substrate analog, product analog, transition state analog. Right, good. Let's see. Yes. So for both of them, C is the right answer. Why? That is because for equilibrium constant, enzyme shifts it equally towards the left and right. So the net movement is 0. But it reduces the EA, that is activation energy. This much we know. What about the rest? It stabilizes the transition state of the reaction. That is something that it does. Okay, so the image that we saw, that I told you, it is taken from Lineweaver. So read about it in details. It's given there. Now, transition state analogs, they bind very tightly because the enzymes are evolved to bind to the transition state more strongly. Okay. Right. So now an enzyme shows a catalytic efficiency of Kcat by Km of 9 into 8 molarity inverse and second inverse. Which statement is most appropriate? The enzyme is inefficient. The rate is limited by the product release. The reaction rate is limited by diffusion of the substrate to the enzyme C. Yes, so this is a new concept and I've included this MCQ so that I can tell to you this particular concept. Okay, so there is something known as diffusion controlled enzymes. So what are these? Certain enzymes, they act very fast. And normally you know that rate limiting step is the step which takes the maximum amount of time to happen. Correct? Now, for this particular diffusion controlled enzyme, the rate limiting step is how fast the substrate is coming to the enzyme. Okay, how fast it is diffusing to the enzyme. Because as soon as it comes to the enzyme, immediately the reaction happens. So you can see that the order of reaction is 10 to the power 8 moles per second. So that is crazy, right? It's very fast. Now, uh, diffusion control enzymes are enzymes whose catalytic efficiency is so high that the overall reaction rate is limited only by how fast the substrate can diffuse into the enzyme's active site, which is the rate limiting step and not the catalytic step itself. So each time a substrate molecule reaches the enzyme, it is almost immediately converted into the product. For example, superoxide dismutase, acetylcholinesterase and catalase. So these enzymes are diffusion controlled enzymes. Okay, they act very fast. Now why? If you know about these enzymes, you will understand. Acetylcholinesterase is used for muscle activity and that is why you can understand how fast it needs to act. Right? Superoxide dismutase, this is important for removing ROS, that is reactive oxygen species in your cells. Right, catalase is also important for acting upon ROS. So these have functions that need to be carried out very fast. So they are basically evolutionarily evolved that way. Now there's another MCQ. The bell-shaped pH profile of many enzymes primarily arises because substrate concentration changes with pH, ionization states of active site residues change with pH, enzyme concentration varies with pH, cofactors degrade at neutral pH. Which one do you think is right? B is right. Correct. Right. It is correct. Okay. Now we will talk about allosteric enzymes. Okay. So an allosteric enzyme changes its activity or function in response to binding of a regulatory molecule that you can see on the screen. This tiny this is the active site of a substrate, and this is the curve for a normal enzyme, right? But for an allosteric enzyme, there is another binding site where this effector molecule comes and binds, and this is called its allosteric site, and it is separate from the binding site of the substrate, which is the active site, and its curve is sigmoidal. Okay, so why is it sigmoidal? Because of cooperativity. What is cooperativity? Often these allosteric enzymes, they have multiple subunits. They are multimers, and when when this effector molecule comes and binds to one of these subunits, it starts showing activity towards this substrate, and it induces all the others to also bind to their effector molecules quickly and show activity towards the substrate, or maybe only one effector is involved and all the others are showing activity towards their individual substrates, right? And that is why this curve is allosteric. So this regulatory site known as the allosteric site is distinct from the active site where the substrate binds, and the binding of the regulatory molecule due to allosteric site induces a conformational change in the substrate structure which can either enhance or inhibit the enzyme's catalytic activity. Okay, right. Now we will look at the next part. Now there are these different terms, which is how this allosteric enzyme functions. So first one is regulatory site, which is the site of allostery where the effector molecule comes and binds. Okay, now this can be an activator, it can be an inhibitor. Then there is a conformational change that is happening in the enzyme. Binding of this allosteric modulator induces a conformational change in the substrate structure, altering its catalytic activity. Okay, this can also be positive or negative change. Now comes cooperativity. So allosteric enzymes often exhibit cooperativity, where binding of a molecule to one subunit affects the binding or activity of the subsequent substrate molecules or enzyme subunits. Multimeric structure, always remember, they normally have multimeric structure. And feedback regulation. So what is with this feedback regulation? So allosteric enzymes often participate in feedback regulation where the end product of the metabolic pathway regulates an enzyme earlier in its pathway. Okay, so they are all interrelated. Now we have the R state and the T state. So kinetics of an allosteric enzyme. So measured activity reflects both R and T state, leading to sigmoidal curve. Okay, activators push towards the R state and inhibitors push towards the T state. So T is tensed and R is your relaxed. Okay, so inhibitor is pushing it towards the T state, and activator is improving its activity, that you can see, right? So from this curve it is becoming either like it is rapidly increasing or decreasing. Okay, now we will talk about allosteric enzymes. Okay, so there can be competitive inhibitor where the inhibitor comes and binds to the one second. Yeah, so there is competitive inhibitor where the inhibitor comes and binds to the active site, right, where the substrate binds. So then that is not allostery, correct? But in allostery there can be non-competitive inhibition and there can be uncompetitive inhibition. So a lot of people get confused about this. Let's try to understand this very nicely today. Okay, So in non-competitive inhibition, what happens is inhibitor binds to an allosteric site of an enzyme substrate complex, causing inactivation. So already enzyme and substrate are bound, and then the inhibitor is coming and binding to the complex and preventing any more further change into the product. Uncompetitive inhibition, inhibitor binds to an allosteric site on the enzyme substrate complex, causing inactivation. Now there is a very subtle difference. We will look at the difference now. Okay, right. So for non-competitive inhibition, you can see that the inhibitor binds to the enzyme or enzyme substrate complex at an allosteric site. Catalysis is prevented due to change in the enzyme conformation. Understood? Okay, great. For uncompetitive inhibitor, the inhibitor binds to the allosteric site of only the enzyme substrate complex but not to the free enzyme, and the inhibitor is not structurally similar to the substrate and does not compete for the active site. So here no conformational change is happening in the enzyme due to the binding of this effector molecule, inhibitor molecule. But in case of non-competitive inhibitor, there was this change in the conformational um, conformation. Okay. Now plots in enzyme kinetics, very important topic and places where students get confused. Okay. So since Vmax is achieved, and as you can see that Michaelis Menten curve is shown there, right? Then Lineweaver Burk plot is also shown there. So since Vmax is achieved at infinite substrate concentration in Michaelis Menten curve that you can see on the left side, it is impossible to estimate Vmax and hence Km from a hyperbolic plot. Right, that is why people came up with this Lineweaver Burk plot where you can exactly see the values of 1 by Vmax and 1 by minus 1 by Km, and so the definite value of Km can be found from here. Now, because of this difficulty, the Michaelis-Menten equation was transformed into an equation for a straight line, which is by Lineweaver and Burk. Now, Lineweaver-Burk plot or the double reciprocal plot is a graphical representation of Lineweaver and Burk equation. Why double reciprocal? Because 1 by Vmax and 1 by Km, minus 1 by Km. Right, so V is the initial velocity and Vmax you know, S substrate concentration, all of these things you know. Okay, good. Now we go back to that equation initially that we had. Now assumptions. There are certain assumptions in Michaelis-Menten equation. Now, this is also very important that you understand, right? So there is no product formation at the start of the kinetic process. That is one. Next, therefore, as long as we monitor the initial reaction rate, we can ignore the reverse of EP going to ES. That we have ignored. That I told you, right? Then assumption 2. During the reaction of an equilibrium condition is established for the binding and dissociation of the enzyme and substrate. That is Briggs-Haldane assumption. Thus, the rate of formation of the ES complex is equal to the rate of dissociation plus breakdown. Okay, so ES complex overall remains constant through time. Okay, because rate of formation is equal to rate of breakdown. So during the reaction, an equilibrium condition is established for binding and dissociation of the enzyme and the substrate. That is called Briggs-Haldane assumption. Now, the amount of enzyme is much less than the amount of substrate present. The enzyme is a catalyst. It is not destroyed. It can be recycled. Only small amount of it is required. Now, the amount of substrate bound to an enzyme at any given moment is small compared to the free substrate. So, this follows that ES is much less than total amount of substrate and therefore S is constant. Total concentration of the substrate is taken constant during the course of this analysis. Okay. Now, only the initial velocity of the reaction is measured. Your amount of P initially is 0. S is much more than S initial. Okay. So, the amount of substrate is much more than substrate initial. And then you have E total is equal to E plus ES. This much we understand. Correct? Okay. Right. So there are a lot more things about Michaelis-Menten equation. Y Km is equal to half of S. That entire derivation is shown for you. And Vmax is equal to K2 into E total. Okay. V is equal to, this is the Michaelis-Menten equation. And what about Km? So basically Km is equal to K minus 1 plus K2 by K1. So that is the rate of breakdown of ES divided by rate of formation of ES. Right? So Km is similar but not exactly equal to, so K, yeah. Okay. Right. So if you can see that K1, Km is similar but not exactly equal to dissociation constant for Kd. Okay, Kd for ES complex. Now K1 is much more than K2. Then Km is much more than Kd. Due to this similarity to the expression for Kd, a low value of Km is interpreted as high affinity of the enzyme for the substrate, and a large value of Km is often interpreted as a weak affinity of the enzyme for the substrate, and that is why you understand this relationship between Km and Kd. So read about this in details. Okay, one small class is not enough to explain all of this to you. So I am just explaining whatever is required for today's class and the MCQs. But for your understanding, you have to read about this in proper details. Okay, and okay, so we'll carry on with the next part of the topic. Okay, so Michaelis-Menten equation, you can see that when there is no inhibition, we have this curve. So, and then when there is competitive inhibition, you can see that Km increases, Vmax remains unchanged. Remember this, this is very important. For non-competitive inhibition, Km remains unchanged but Vmax decreases. Okay, so inhibitor reduces the enzyme activity but it does not bind to the active site. It is not changing the substrate's affinity towards the enzyme, right? It is not affecting that. That is why Km is remaining unaffected. Now, interestingly, for competitive inhibition, we saw that Km increased, Vmax is unaffected. For uncompetitive, both are reduced. Okay, and for non-competitive or mixed inhibition, we have Km is unaffected and Vmax is reduced. Now, what is mixed inhibition? Read about this and tell me. Okay, it is also very similar to non-competitive inhibition. So, in non-competitive inhibition, you saw that it could either be binding to enzyme or enzyme substrate complex. So in mixed, it is like something similar happening. So you can read about this. Okay. Now we will try to solve a few questions and then the class will end. Okay. So when the velocity of the enzyme activity is plotted against substrate concentration, which of the following shape is obtained? So remember, velocity against substrate concentration. So this is the normal graph that you see. B is hyperbolic. Good. And what about the second one? When kinetic property distinguishes allosteric enzymes from classical enzymes. Yeah, great. So you're saying that it is sigmoidal. Let's look at the right answer. Yeah, so it is B and B, right? So this is hyperbolic for normal enzymes and it is allosteric for allosteric enzymes. So it is sigmoidal for allosteric enzymes. Now allosteric enzymes show cooperative substrate binding, producing a sigmoidal or S-shaped curve rather than hyperbolic curve of Michaelis-Menten equation and Michaelis-Menten enzymes. Now we have classification of enzymes explained in one single question. Okay. So now there are different classes of enzymes. There are total 6 classes of enzymes. And what are they? Okay. So you can try to match these. There are ligases, there are liases, there are oxidoreductases, hydrolases, all of these different transferases, right? So all of these different classes of enzymes, you can try to match which is which. D. Okay, let's see. Yeah, so D is the right answer. You can see that ligases uh, is used for catalyze the reactions in which two chemical groups are joined using ATP. Right? So we have T4-DNA ligase, we have E. coli DNA ligase as examples. Then there is lyase. So lyase involves add water, ammonia or carbon dioxide across double bonds or one second. Two is four, yeah. So hydroxylase is adding water across a bond and hydrolyzing it, right? And what about the next one, which is oxidoreductases? This you know that they act to either add or remove hydrogen atoms. And what about lyases? They're adding water, ammonia, or carbon dioxide across double bonds or remove these elements to produce double bonds. Okay, great. Now we have two more questions. Try to answer them. In which form the enzyme exists when the maximum velocity is observed? Okay. And in enzyme kinetics, the turnover number is defined as maximum number of substrates that can be converted to the product per mole of catalytic site per second, concentration of enzyme required for each. B. Yeah. And what about the next question? A. Right, right. Good. So let's see. Let's look at the right answer. That is correct. So B and A. Okay. So the enzyme would be as ES for maximal activity, and A would be the right answer that defines turnover number. Okay. Now, which of the following is true regarding Michaelis-Menten equation? Km, the Michaelis constant, is defined as the concentration of the substrate at which the enzyme is working at its maximum velocity. It describes single substrate enzymes. Km, the Michaelis constant, is defined as a dissociation constant of the enzyme substrate complex. Right. Yes, that is true. So all are wrong except B. Okay. So Km is defined as the concentration of the substrate at which the enzyme is working at half of its maximal velocity. It is also a measure of the affinity of the substrate for its affinity of the enzyme for its substrate. And Michaelis-Menten kinetics assumes non-covalent binding between the enzyme and the substrate. Okay, and it is useful only for single substrate reactions. Okay, now we have two more questions. Which of the following is not a characteristic of enzyme? Enzyme accelerate reactions by increasing free energy of activation. They often bind to the transition state of the reaction better than the substrate. Enzyme inhibits saturation kinetics. Enzyme works at optimal temperature and pH, which is not a characteristic of enzyme. Very direct question. A. Yeah. So small organic molecule bound to the apoenzyme by covalent bond. Okay. Good. Let's see. Yeah. So that is correct. Now remember that when the substrate, you remember in induced state, the substrate changes its conformation and it becomes exactly like the active site. Now it is called transition state, and the enzyme binds to the transition state very tightly. Now enzyme accelerates reactions by decreasing the free energy of activation. So A is wrong, and the next one you can see that prosthetic group is the right answer. So when okay, so coenzymes, they are loosely bound, but prosthetic groups, they are very tightly bound, and they are often permanently bound with enzyme. But coenzymes are only temporarily bound, and cofactors, but they are organic. For example, FAD, heme, and biotin. Right, so NAD plus and COA, these are organic cofactors, and they are normally loosely bound coenzymes. Right. So now we have one more question. The above diagram shows a lock and key model of the enzyme that has been affected by an inhibitor. What do you think is going on here? What do you think is going on? Uncompetitive inhibitor, competitive inhibitor, non-competitive inhibitor, allosteric inhibitor. B, competitive. Yes, that is correct because inhibitor is binding at the substrate binding site, right, at the active site, and that is only possible in case of competitive inhibition. Next, there is a graph. The given Lineweaver Burk plot for an enzyme shows that that has been affected by an inhibitor. So what is happening here? What form of inhibition do we see here? Competitive, non-competitive, uncompetitive, and mixed inhibition. So what is happening? With inhibitor, without inhibitor. Okay, so C is uncompetitive. That's correct. It is uncompetitive inhibitor. Right. So now we have a few formulae here and there. So in an enzyme catalyzed reaction, dash provides information on substrate binding and dash provides information on the reaction rate. Which of the following equations is used in Lineweaver Burk plot? C. Okay. And D. Great. Let's see. Yeah, that is correct. Perfect. Okay. So how much do we have left? We are almost. This is the last topic for today. Okay. Right. So we will go and complete this as quickly as we can. Now, for an example, RNase A is given to you. Okay. Now, RNase A is an enzyme which cleaves single-stranded RNA. How does it do it? It uses two different steps. So, RNase A cleaves RNA to form 2,3 cyclic phosphate intermediate. And the intermediate hydrolyzed to produce 3' phosphorylated RNA fragment. In its reaction center, there are 3 residues that you can see. Okay. Now, one of the residues is histidine 12, which acts as a general base, and histidine 119 acts as a general acid to promote nucleophilic attack and bond cleavage. Okay. Now, this histidine 12 again acts as a general acid, and histidine 119 acts as general base to promote hydrolysis. Right? So, that is how single-stranded RNA gets cleaved. Now, interestingly, you can see that the bond cleaved is actually phosphodiester bond. Specificity is it often cleaves after pyrimidine bases, that is cytosine and uracil. And the catalytic residues are 12 and histidine 12, histidine 119 and lysine 41 in the active site. Reaction mechanism is a two-step acid-base catalysis reaction. The intermediate formed is 2', 3' cyclic phosphate intermediate, and the final products are RNA fragments ending with 3' phosphate. No cofactor is required for it. That is why it is so difficult to work with RNA, because firstly, RNases are present ubiquitously everywhere, right? So even if you boil water up to 100 degrees centigrade for let's say 1 hour, there can be active RNases in it. That is why water is autoclaved twice. So it under very high pressure, very high temperature, it is autoclaved twice to remove these RNases. Plus they do not require any cofactor, right, to get activated. They are active at every temperature. So that is why it's very difficult to work with RNA in the lab. Now see. So RNase A catalyzes the cleavage of phosphodiester. Okay, so you're saying that it is D. Let's look at the right answer. Yeah, so D is the right answer. So in RNase A, catalysis, all these two are important residues and they have the ability to both accept and donate electrons. And specific substrate for RNase A is cyclic CMP. Okay, so that's it for today's class. Thank you so much. These are the different references that you can go through. And then we have the last thank you slide. So thank you so much for your attention. Yes, I'm pursuing PhD. No, we don't have to write a book. You can write book chapters. So if your PhD guide or supervisor, he is invited for writing book chapters, then you have to write. Otherwise, your thesis will be like a book. So whatever research you have done, that will be kind of like a book. That is a thesis. Plus, you will have to publish a lot of papers. So novel things you will have to, in which topic I'm doing PhD. So my PhD topic is protein engineering and structural biology. Okay. So whenever I'm talking about proteins, I get more excited and more because that is more familiar to me rather than other things. Okay. So if that is all, then we can end the class for today. I'm having fever. Okay. Have a good night's rest and then we can continue with the next class again. Bye bye. Yeah. That is why I'm